We added one speaker to our program, which is Dr. Matthias Rath. He's the founder of our research institute and of the concept of cellular medicine. He will add a few things to the presentation of John Chia. Welcome, Dr. Rath.
Thanks, Mark. Goedenavond. The reason why I'm speaking here is something very exciting, and you all found this publication on your table and with 3 signatures on it, and you may have asked the question, what is the significance of it? As all publications in science, they have a certain tone and a certain content which doesn't immediately open up to everyone because of the technical language. So let me take a few moments to guide you through the significance of this paper. It has a lot to do with my life and with my scientific career, and secondly, since many of you are therapists, and the questions came again and again, what does it have to do with the patient, with the human system? I will try to answer some of those questions as it relates to cardiovascular disease. When I started this whole field of research myself about 30 years ago, the questions were the same than they are today. We essentially didn't know what cardiovascular disease is. The problem is, when you go into the established institutions today, the same questions remain. No cardiologist in Maastricht or Amsterdam or New York or Vladivostok can explain these 3 questions to you or give you answers to those questions. First, why do we get infarctions of the heart but rarely of other organs? If cholesterol, as they tell us, high cholesterol damages the blood vessel walls, thereby initiating atherosclerosis, we would get atherosclerotic plaques not just in the coronary arteries, but along the 80,000-mile-long pipeline making up the human body, in the veins, in the capillaries, everywhere. We would not get just infarctions of the heart, but of the nose, the ear, the kneecap. No one ever has seen anything like that. Second question, why do we get infarctions in the artery, arteriosclerosis, but not in the veins? And why do animals get heart attacks, but people, why do people get heart attacks, but animals don't? Heart attacks or atherosclerosis in animals is not impossible, but it just does not occur naturally as it does in humans. And there's a law in medicine and biology that says an epidemic continues unless and until the true cause is found. In other words, we shouldn't wonder why every second person in Maastricht and the Netherlands and the industrialized world dies of a disease if we don't answer the basic questions of this disease. "30 years ago, those 2 chaps made history by getting the Nobel Prize in Medicine and Physiology, Goldstein and Brown. And they became like the pioneers of the philosophy that high cholesterol, or the cholesterol-carrying molecule, low-density lipoprotein, or LDL, is actually the culprit. These pictures show in a nutshell what we're talking about. The lipid and cholesterol core is here, cholesterol triglycerides are there. There's a protein that stabilizes this globule, and it forms the lipoprotein particle here. My research started with a variant of LDL. in short, it is the same particle you just saw, lipids, cholesterol in the center, a stabilizing protein to give it a round spherical shape. And this variant, called lipoprotein little a, has a sticky macromolecule, a huge protein wrapped around. The significance of this particle was completely unknown at the time, and together with my colleagues in Hamburg, we looked into the question, what does this variant of LDL has to do with human atherosclerosis? At that time, the structure of this violet molecule, this sticky macromolecule, became known, and it was homologous to a factor that you know from the clotting fibrinolytic system, which is plasminogen. And that sticky outer protein made this LDL variant called lipoproteinase stick to essentially everything. These are just three of the most important molecules that affect the atherosclerotic process. Fibrin, so lipoproteinase sticks to fibrin, it sticks to collagen, it sticks to endothelial cells, and about a dozen of more substances in the blood vessel wall. So it was no surprise that when we studied the composition of human atherosclerotic plaques from patients at the University of Hamburg, that wherever we found the LDL particle, which is the fat-containing LDL particle, we also found the sticky protein. As you can see, there is exactly co-localized. These are serial cross-section through a coronary artery plaque. The brown color marks the protein for LDL. The violet color marks the additional ApoA protein. What that picture tells us is that the researchers that tried to understand the nature of human atherosclerosis or the composition of the human atherosclerotic plaques and came to the conclusions it is LDL, they just didn't look carefully enough. Had they looked also for the sticky protein, they would have identified it's not LDL, it is the variant that has this additional glycoprotein. The consequences of this little picture here are dramatic. Considering what we now know, that there is a $40 billion statin industry that tells the people we need to lower LDL and you prevent heart disease. And LDL has very little, I wouldn't say nothing, but very little to do with human atherosclerosis. Plus, statins do not lower lipoprotein A. They lower LDL, but they do not affect this risk factor, LPA. It gets even more significant when we then studied, again, these are studies 30 years ago, when we studied how does this lipoprotein A, the sticky LDL variant, relate to the development of atherosclerotic plaques, we found that actually the larger the plaques are, the more lipoprotein A is in these plaques. In other words, lipoprotein A deposition contributes to the growth and ultimately to the development of stenosis, both in coronary arteries as well as in cerebrovascular arteries. In the meantime, there is no question that lipoprotein A is the leading lipoprotein or lipid factor independent of anything else contributing to cardiovascular disease. It is the primary risk factor for heart attacks, coronary artery disease, for stroke, cerebrovascular disease, for peripheral vascular disease, for restenosis after bypass surgery, and angioplasty. If you want to have the reference papers, you go to the website of this symposium, and we collected them for you there. So 25 years after it was clear that it's not LDL, but it's lipoprotein A that makes up the human atherosclerosis that causes heart attacks and strokes in millions of people, the European Society of Cardiology finally got around and said, well, we better recommend the screening for lipoprotein A, at least in patients at intermediate or high cardiovascular risk. And here comes the exciting part of it. This is a discovery I made in 1987, so that's 28 years ago. At that time, it was known that lipoprotein A was present in men, in Homo sapiens, and subhuman primates, but not in other species. It's a fascinating molecule. And I said, oops, isn't there something else that is specific to the human system or to subhuman primates? Of course, it was the inability to manufacture vitamin C endogenously. So there was an inverse relationship. We people cannot manufacture vitamin C, but interestingly, we manufacture a very dangerous molecule, lipoprotein A. Vice versa, animals produce high amounts of vitamin C, between 5 to 20 grams per day compared to the human body weight. But they don't need lipoprotein A, so it doesn't occur in their metabolism. So what was the significance of that? Well, I talked to a Nobel laureate in Texas, whom you've seen before, Michael Brown, and he said, you're crazy. So that was that. Then I talked to another Nobel laureate, Linus Pauling, and he said, you better come over and join me. So we solved this puzzle, which I did, and it changed my career. I left the work at the German Heart Center in Berlin, and I said, this is so important that if I don't do it, no one will, because vitamin C or vitamins were ostracized at the time. So lo and behold, in 1990, Linus and I published a paper in the Proceedings of the National Academy of Sciences, basically saying in humans and subhuman primates that suffer from this genetic defect of not being able to synthesize vitamin C, being prone to scurvy, there was the need for a repair factor popping up in evolution that mends the arteries in times of scurvy. Scurvy being in Dutch, scherboid, yeah. The thing that the sailors, you were sailing nation, so lots of your ancestors died from that condition on sea. So during those conditions, there was a tremendous advantage of having a repair molecule right in your body that immediately mended it when you had lived at danger of this disease. But as you can see from the title of this paper, it had this word hypothesis there, right there, which means we had a fantastic concept, made a lot of sense, made so much sense that we sat down and wrote this paper, but the proof was very limited. The paper basically outlined that there are two ways how arteries are kept stable. First, you have enough of vitamin C. Vitamin C is needed for the production of collagen. So there are intact and sufficient collagen molecules that give the artery walls sufficient stability. Or you have no vitamin C synthesis in the body, you get too little vitamins in the diet, and you suffer from hypoascorbemia or vitamin deficiency. In that case, first thing that happens is collagen is underproduced. The stability molecules of the body and the blood vessel wall are not there in sufficient quantities. In that situation, a molecule that is a fantastic repair factor, lipoprotein A, has a dramatic advantage because it is being deposited in the impaired, weak muscular wall. Is that so far clear? If you can't follow, just say. So now we suddenly understood the function of this lipoprotein A molecule. which in conventional medicine was essentially demonized. They said, oh, you have this factor, you are at risk, there's nothing you can do. We need to, case the lipoprotein level is too high, we need to put you on apheresis or dialysis for lipoproteins, as it is called. Well, the role of this lipoprotein molecule in nature is it's a formidable mobile repair molecule that uses LDL as a transport vehicle, and the adhesive apoprotein A molecule is a substitute for collagen. In other words, the length of this sticky protein is twice as large, in some cases 3 times as large as a collagen molecule. So if collagen breaks down and you have a mobile repair molecule, it's a fantastic solution of nature to counteract scurvy and blood loss and death. And of course, the consequence of that is that we suddenly also understand the nature of the atherosclerotic plaque, which was a God-given event, still is. The atherosclerotic plaque is nothing else than a life-saving repair process for the artery wall when it is weakened by deficiency, most prominently in vitamins and other micronutrients. And when this repair process goes on for too long or overshoots, then the plaque develops. Once again, you open up a textbook of cardiology today, they talk from page 1 to page 2000 on plaques. But at no line, in no paragraph, is there a definition what it is, why it occurs, what is the nature of this. They tell you that lipoproteins contribute to the plaque development in the following way: that LDL just damages the lining of the artery wall, and by doing that it enters the wall, and in the wall it is beaten up by macrophages, police cells. And when these police cells overeat on cholesterol and LDL, they die, and they spit out the cholesterol in the wall, and that's the plaque. What a nonsense. Why would nature do something like that? It makes absolutely no sense. It's absurd. It's fatalistic. What makes sense is if there is a weakness of the artery wall, that repair molecules go in there. They are not destroyed. They are in there deposited intact because they're needed intact to repair the wall. And the consequence of that is, of course, that this new concept that we have been presenting is not a fatalistic process. It makes sense. It's an absolutely necessary regulatory mechanism of nature. And most importantly, most significantly, if that's the case, then we have to postulate this process is reversible. It's not a one-way process. If you fill up, if you counterbalance it with micronutrients, because they're caused by micronutrient deficiency, then this process must be, by the laws of nature, must be reversible. So we have prepared an animation. I just can show you the still shots of it, but atherosclerosis starts not with an overload of lipids that damages the wall, but it starts with those gaps here, crevices, developing between the endothelial lining. It starts with the endothelial cells moving apart because of vitamin deficiency and no longer closing. They are closed normally like this, and with vitamin deficiency they open up like that. So here you see the plaque being the repair and beginning to overshoot. And here you see that in principle it's reversible when you regenerate, when you re-establish the underlying stability with the help of vitamins and micronutrients. And of course, the fascinating conclusion of this new understanding is that we suddenly understand all questions that I raised at the very beginning. Why do we get infarctions of the heart and not the nose? Very simple, because the heart constantly moves. And the coronary arteries riding on top of a moving organ, they are exposed if they are not stable. So the mechanical stress from the pumping heart explains the coronary arteries as the primary location of cardiovascular disease. We understand why we get atherosclerosis, because the blood pressure in the arteries is 100 times higher than in veins. And we understand why animals don't get heart attacks, because they manufacture their own vitamin C and we humans can't. There was the question, is there proof in the human system? Dr. Niedzwiecki mentioned the study. It's published in the book that you have in front of you in its entirety in the annex. Nutritional supplement program halts atherosclerosis is the title. Well, it's not just holding it. This is a good example that it can actually reverse existing coronary deposits. On the left-hand side, you see the left coronary artery of a 51-year-old patient. The X-rays are made with computed tomography, and the right-hand picture shows you the same patient, same heart, same location 1 year later. After 1 year on micronutrients, there is no more deposit. So we are able to prove, this is, by the way, the first time that something like that has been shown in the history of medicine, that there is a natural reversal of coronary artery disease possible. So with no drug or with no procedure has this been shown to be the case. We thought this is it. Dr. Niedzwiecki and I, we went to Washington, D.C. to have a press conference on this study, because we felt that, well, what else do you need as a proof? Well, we were very wrong. That was just the beginning of the battle to prove things. The ultimate proof needed to come from something that we couldn't prove in the human system. In other words, we needed to induce scurvy and ask the question, does lipoprotein A enter the wall in that condition? But since there existed no animal model where we could study that, we needed to create this animal model. And that was Dr. Niedzwiecki's belief in this concept, why she talked with John, whom you've heard before, to establish exactly that model. And John, by the way, he worked for 5 years to establish this model that he just presented to you. But that model is the only way how we can prove, conclusively prove, what you just heard. Oh, there was a question also on, is enough or is more clinical evidence available? We summarized some of these studies on our symposium website. Here's just one of them, analysis of 9 studies with almost 300 participants over 10 years. Vitamin C intake above 700 milligrams resulted in a 25% decrease of cardiovascular disease risk. There are many more studies, but that's just one of them. So, but going back to this animal model, so the necessity to prove the nature of human cardiovascular disease could not come from humans. We needed to create an animal model that mimics the 2 key metabolic factors from the human system: the inability to produce vitamin C and the ability instead to produce human lipoprotein A. So this is genetically modified. Actually, it's a knockout plus a new insertion of human genes into this animal model. So as you can see, this is what you know already. Animals produce their own vitamin C, no LPA. Humans produce no vitamin C and do LPA. Animals, they produce their own vitamin C and no LPA. And here we produce an animal that is like humans, as you can see, with these 2 factors. No vitamin C synthesis, and instead LPA molecule synthesis. Is the setup of this animal clear? And John showed those experiments, but just to repeat, on the left-hand side, these are artery walls of animals that did not feed or did not get high cholesterol in the diet. The only thing that happened with them is we omitted vitamin C, and these plaques developed, proving that the plaques are actually repair processes. And on the right-hand side, you see that with optimum vitamin C, there's no need for this repair process. And then, of course, the pictures that John showed to you, that we detected lipoprotein A molecules inside the wall. I don't have time to go into all aspects, but maybe the most significant and most, for me personally, the most fascinating aspect of that is from genetic analysis, we know that the moment when vitamin C synthesis got lost in our ancestors was about 40 million years ago. And we also know that it was exactly that time when lipoprotein A popped up. So this animal model actually reproduces a moment in time in the metabolism of our ancestors that led that we are here today. Without LPA, our ancestors would have died out from scurvy. No question about that. And understanding this evolutionary event, this switch 40 million years ago, suddenly explains to us why we all suffer from cardiovascular disease, and every second person in this city and in this country will die from this disease if we are not intellectually capable of demystifying this problem. And the significance of John's work and Dr. Niedzwiecki's work, and a little bit my ideas some time ago, is that from now on, from this week on, we understand this. Mankind knows what has happened then, and by reproducing what has happened then, we can conclusively prove the nature of human cardiovascular disease. Am I reaching you? And that was the reason why we felt not only should we put the paper on your table and sign it, because we feel this is a historic moment. We also showed to you the press release because we want the whole world to know that this is the moment. And why are we not at the convention of cardiologists presenting this? Think about it. What we are doing here is we are giving you, many of you are natural therapists, a weapon into your hands, an intellectual weapon that can emancipate this entire field of natural health by providing it with a scientific base. Emancipate beyond the level that is already available from the experience you have, from the individual ingredients that have been tested. This here is a concept that only you can answer. It is not the statin story. It is understanding regulation of nature, or natural regulation. Some of you may ask, well, was it so easy? Not really. over the past 15 years, the lobbyists of the pharmaceutical industry, they filed more than 100 lawsuits against Dr. Niedzwiecki, myself, the Institute. No time in history of medicine was a concept so fought than this one, than cellular medicine or cellular health. This picture you see on the left-hand side is a picture from the cellar of our attorney. The files that he collected over those 10 years are 500 meters long, half a kilometer. That tells you how furious the status quo is on what we just told you tonight. And the fact that we can do this here and that I'm not sitting somewhere in some remote location, and why do you think that is? Why are we here tonight? Why can we share this with you? What do you think? How do you think we were able to defend ourselves in those lawsuits, in these courtrooms on 4 continents? How do you think? Evidence. So if there's any doubting soul in this room tonight that what we told you, whether this is heart disease or cancer, or whether it's Dr. Gotch with her Lyme disease work, that's the answer to that. And I think we cannot be clear enough on that. Dr. Niedzwiecki told you that the plan is, they already calculated it. They want to make $47 trillion. It's 42 billion euros in the next 15 years from these diseases. They want to take that money from you. Now that amount that I just mentioned is the gross domestic product of the entire world, except for 2 nations. So that's the dimension. But they can do that only if we allow them. And this is why it is important that we have this meeting. It is important that we talk clearly to you. Whether you have remaining questions or doubts, we need to say the things like we see them. With this new scientifically proven understanding of cardiovascular disease, the natural therapists, many of you here, have a chance to take the scientific lead in the control of the #1 health problem of our time. That's the significance of the publication in front of you. Take the press release. Here it is in English, but I think we also have a Dutch version, and work with it in your community with the local newspapers. Wherever you can, send it to people who you know. And 2 days ago, we had the meeting here in 1 floor higher, and we decided to revitalize the call that Linus and I issued in 1992, 3 years ago, when we had just a concept, no proof. And we said now that the time is here where we can prove this. It's time to revitalize this call and give it the name Maastricht Manifesto. I won't go through all of it, but well, let me insert one thought here, and that is this one. I was thinking how I could illustrate what we are doing now, what this means here. And the closest one was this one. I'm not comparing myself to this man here, make it very clear, but the significance for human health is comparable. There was a time when mankind was haunted by infectious diseases, and the status quo was telling, well, that's a curse of heaven, because you are all sinners. Until this man came and said, well, wait a minute, maybe another reason, maybe microorganisms. He was ridiculed. He was fought for 25 years. This is a caricature. There were cows growing out of the arms of people who have been vaccinated with cow vaccines. And it was 25 years later until everything was accepted when he vaccinated a 9-year-old child against rabies. 25 years, it's exactly the time that we are talking about. That's the end of this call. With millions of lives each year at stake, no time should be lost. That's the handwriting of Linus. So we have, if you want, we invited him to join us from the grave. But it's as present as today. The need is as pushing, as concrete, as urgent, as essential as it was then. And we didn't say we are the greatest. We said there is so much compelling evidence that now we need to encourage everyone around. And I'm encouraging every person here who is a therapist, who works in this field, to join this effort, to now conclusively work along those lines. If we can use our knowledge, get the evidence in patients, do studies, observational studies, if we cannot do larger ones. Because that's the dimension, the goal of eliminating heart disease as a major cause of death and disability is now in sight. When you go out, you will have the possibility to pick that up. I'm at the end of my little intermezzo here. It was important for me to use your presence here tonight and talk to you very clearly. Thank you for your attention.